unmute. Right. Just camera here. Good.
Hi, everyone. Can everyone hear me online? And also here in person so that I'm not too soft. Okay, good. So thank you everyone for coming. So we are really, really excited to have representatives from Altium and Cytosurge with us. So they're going to talk about precision live cell, single cell sequencing. And this falls under Panoramics Vision Enlighten-me chapter, the Spatial Nexus program. Just briefly, at Panoramics, we are very much motivated to bring in new technologies because, like biologists, we have different types of questions, we have different needs. And in order to adopt, we believe that education and understanding of the technologies are actually paramount to actually revisiting what kind of experiments and what kind of tools we need. So without talking too much, that's the purpose of the Spatial Nexus program. We had True Not Spatial, so we're really busy and we couldn't really invite too many technologies and platforms for this year. But we're really excited to kickstart this with Cytosurge and Altium, who's going to be presenting their precision single cell using live cells. So I'm going to let them present and introduce themselves and then to take off the presentation. So a warm welcome to them.
Thank you so much, Shani. Oh, I have to. I'm too tall, I guess, for the camera. So yeah, if I walk back, it works. Thank you so much for being here. And so I have Barry from Altium with me. She is our US East Coast representative. Please go and talk to her if you want to know more about Altium and what they can offer. And just briefly, for me, it's really rare to be in this room here. I did my postdoc over at the Lunenfeld and we had lab meetings here and it was always very, very intense. Usually it lasted 2.5 hours. And yeah, it's kind of cool to be back. So what I want to do now is I want to talk to you about our technology and what you can do with it. Just make sure that I can find it again. Here we have our device. So I'm working for Cytosurge. I've been with the company for 6.5 years now. So I'm not one of the founders, but I know the founders personally very well. And we've been talking about how to use the technology in biology well before I joined the company. And what I would like to do now is kind of to show you the flagship applications in single cell biology and also kind of try to show you the versatility of the device. So without further ado, I'll start talking about the technology. What you can see here is on the right-hand side, we have the core of our device, the nanoneedles, the instrument, and you can see that we put an incubator on top. So I just showed you that you can kind of imagine how it looks like. And we usually start a conversation when I present with the following slide. So as a biologist, you all know EMT, epithelial to mesenchymal transition, an essential pathway or kind of morphological morphing pathway in early development. So the primitive streak, for example, but also in cancer, when it goes wrong, this is really where it turns a cancer turns bad. What you can imagine is you have your epithelial cells, they start to go over to a more mesenchymal state and become more invasive, but it's not like a snapshot of a state. It's fluctuating between all these different things. And if you really want to understand it, what people do now is they use regular single-cell RNA sequencing. So they take snapshots at distinct times. And this is great to kind of find who is there in the year, make sure what's going on. But you have one major issue. So once you've taken the cell to measure, it's destroyed. You cannot do anything else with it. It's gone. And this is exactly where we come in. So because of this destroying the cells, you don't have the ability to track dynamic responses. and you rely on inference correlation to find the causal links. And I like to ask you, so what if you could go take a sample from your cell without killing it, record the whole transcriptome, and sample the cell again after a stimulus? So you have two time points that are linked together. And this is exactly what you can do with our technology. At the core, we have the so-called nanosyringe, so the smallest syringe in the world. We have an opening here that is roughly between 600 and a couple of more nanometers. That's not working. Okay, try it again. Yeah, so you have this nanosyringe, an opening of about 300 to 900 nanometers. The whole thing is hollow. And what we do now with it, it's also a kind of an AFM cantilever. So what we can do is we can mount it on our microscope, an inverted microscope. You have the cells on the bottom there. You have the cantilever coming down in the Z axis. Once the cantilever touches an object, it starts to bend. We can measure the laser diffraction. So you see here the laser, and then kind of calculate back how much force we put on a single cell. Hence, we can be extremely gentle with them, and we can either inject or extract from single cells by applying positive or negative pressure through our microfluidic system. So we have controlled physical access to the cells. We are very gentle because we have force feedback control. We can either take in the sub-picoliter range from a single cell or inject to it. And the whole geometry here can be adjusted, and that becomes important for the rest of the talk. We can also do other things than injecting or extraction. And we're proud it's Swiss engineering, so it's very precise and stable. So the workflow, I'm going to talk to you first about the so-called life-seq. So you can take a sample from a cell, stimulate it, take the sample again from the same cell, and therefore we call it life-seq. How does that work? Again, we have here these mesenchymal epithelial to mesenchymal transition. We have these cells. We take about 20 cells, poke them, set aside the sample we've taken into a buffer droplet, put it away. Then we stimulate the cells. And because we have recorded the coordinates with live cell imaging, we can follow these cells now over time. And then after 4 or 24 hours, we can go back, poke the same cell again, take a sample, set it aside again for sequencing or other things. Important to know, in this droplet, we already have all the reagents that you use for the library preparation. And the readouts we use is we can look at the RNA, so we can have more or less a full transcriptome from that biopsy. We started to do some proteomics, so we can measure the protein content by a mass spec. And on top, we have the phenotypic appearance of the cell. So we can do bright field imaging, or we can do also fluorescent imaging, and we can record the context. So this comes a bit more towards the spatial point of view. So we can have a co-culture between macrophages and cancer cells. We can correlate the transcriptome of the cancer cell, whether it was in close distance to a macrophage or not, or whether they interacted. And this gives us deep insights into a single cell. So what we can do is basically, instead of only having 2 dimensions in your UMAP, we add 2 dimensions more. So we have the time, because we can follow the cell and record the transcriptome, and we have the phenotype on top. And this whole thing gives you a lot deeper insight on how a cell behaves after a stimulus. To illustrate that, we have here this little video. What you can see here is our device in action. Come on. Yeah, no. Okay, apparently it doesn't like me that much today. Let's see if I can get the video. Oh, now I have a pointer. That's cool. Oh, now I know. Yeah, it's embedded, but I think the issue is now I wasn't it was the pointer and not the clicking. So what we have here is the device. So you saw the operator was kind of clicking on a cell. It's GFP positive HeLa cells. We had on the right hand side we had the parameters that we set. We saw that the cantilever approach, we pierce the cell, and now it goes back to the deposition slide. And what you can see here now is a fluorescent content of the cell that we taken out. And because we know the exact dimensions, we know how much we sampled. So in this case, we took a half a picoliter from that cell. So now we can say, this is interesting, but what can you do with it? Or what is the value of it? So what we have here is kind of one of the use cases that has been published by one of our collaborators. So they had a scenario, they have macrophages carrying an NF-kappa B LPS reporter. So if you stimulate them, they turn red. And when you look, what you would expect is, okay, these are cells, they will react more or less the same. But the thing is, each and every cell has an individual reaction to the LPS. And you can see when you look at the response curve, how much fluorescence you generate. Every cell has a bit, one has more, and you don't know really why. And you can try to address that with single cell RNA sequencing. So having a sample at 0 hours, a second dish at 4 hours, do the single cell RNA seq. And then what you have is something like that. What you can see is that NF-kappa BI kind of positively correlates with the TNF or the response. And you think, okay, yeah, that's a positive regulator. So more NF-kappa BIA you have in a cell, the more response you get. But this is kind of not really what's happening. And when you take now our device and you have a quiescent macrophage, you take a biopsy, record the transcriptome, stimulate the cell with the cells with IPS, do the time-lapse imaging so that you can see how the cell reacts to the stimulus, and then do the data integration. You kind of do not end up with a positive correlation, but rather with a negative correlation. So a cell that has little NF-kappa BIA at the beginning will have a stronger response to the stimulus. Then when you go back to the textbook and start thinking about it, it makes absolute sense. So the single-cell RNA-seq show the positive correlation, and our show the negative. And why is that? As I said in the textbook, when you look at the pathway, you have NF-kappa BIA is the inhibitor of the NF-kappa B pathway. So it sequesters NF-kappa B into the cytoplasm. Once it gets stimulated, it will release, it goes into the nucleus, it binds to the DNA, and then it starts the response. So if you have little at the beginning, the response will be stronger. And why do we have the opposite in the single-cell RNA-seq? It's pretty simple. It's a feedback loop. So kappa B regulates its own inhibitor. So what we kind of see is the regulative feedback loop. So with our device, you can really tease into this mechanism and try to understand what's really happening in the cellular signaling. Then the other thing is kind of looking at cell fate trajectories. So what we have here is adipocyte precursor cells that are differentiated into adipocytes over 2 days. So we take a sample at 0 hours, 48 hours. We also include express barcodes because over 48 hours, it's kind of difficult to follow an individual cell. And what you see here is basically the trajectory of these cells. You see that the brown cells become the light brown cells, and we know exactly that they went from one state to another. So this is basically, we measure the ground truth of this cell fate transition. And if you go now and say, okay, you can do that with single-cell RNA-seq as well, and you use some kind of algorithms that finds the trajectory, that's kind of what you end up. You get a trajectory, but it's kind of either inversed or somewhere all over the place. So we can find things that you cannot really easily do with inference. Next, just briefly to give you a bit of a feeling what we are doing. So we often have clients, they come to us, we want to buy a device, but we need preliminary data to kind of get a grant. So we take their experiment, do a small scale, minimal viable experiment in our house. So what they had is they had cells that they want to measure at 0 hours, do a stimulation, then after 24 hours again. We did like 2 sets of experiments. What you see here is the gene detected in the 2 series and the gene counts. We had the negative controls, which is basically just pipetting the buffer with our device to make sure that we don't have any carryover. Then we had the positive controls. What you see here is on the positive controls, what it was 0.2 picogram of total RNA and 10 picograms. So you can see we can detect quite a few genes. And then you have the treated and the untreated cells. They are definitely above the negative control, a bit lower than the 10 picograms. So we are somewhere in the range of probably 2-3 picograms of total RNA, that kind of what we have. And that's kind of what we see. We see that the experiments are more or less reproducible. And what we did then is kind of seeing, okay, what are these cells are doing after they've been stimulated? So what you can see here is the PCA plots. You see in the middle are the unstimulated cells, and they take 2 routes. And they kind of go towards different fates. And this is kind of cool to see that, because you really follow the individual cells, you know the trajectories. And when you look at the genes expressed, in the middle, you have the control cells, the unstimulated ones, and then you have the other ones, the 2 fates going out that are quite distinct. And these are kind of things that you can do with the device. You really understand where a signal cell is going. Most of the people then ask the next question, okay, what kind of throughputs do you have? And this is kind of the Achilles heel of the whole thing. The initial paper that I showed you was done with a so-called AFM add-on FluidFM. So this is the older, less capable brother of the current instrument. And when you look at that figure here, I would say, so they did the library preparation with this homebrew SmartSeq 2, and only about 40% of their libraries were successful. The sequencing was also Illumina-based, and to get that figure here, I'm pretty sure this took about 6 to 9 months. And it's also highly dependent on the operator. Now we took that workflow over to our device, and that's how it looks like. And we have more access to the field because we have a more working distance or working XYZ stage. Library preparation, we do Luthor HD. So we are up to more than 60% of these really tiny amounts of RNA give us a good library. Sequencing is the same. And we cut down the data collection for a figure like that to at least 1 or 2 months. So that's already 5x. And basically everybody can do it. It's really easy to use the device. And one of the things we did to make it more easy is we started having automated tracking. So you can see here the tracking function of our device. So you see here, you have the coordinate of cell 20. That's the cell that's marked there. And now we can just go and see it, how it moves around, so that we can make sure that we see the same cell. This is kind of neat. So this cell was already poked, and after 4-6 hours, it will start bubbling up, and it's not dying, it's dividing, kind of showing that we did not have a huge effect on the viability of the cells. Now I think, yeah, now it starts dividing.
Could I ask a question?
Sure.
So I'm going to ask the Werner Heisenberg question. They're actually observing those change results. So have you done exhaustive control to show that by poking the cells and so forth, you don't probe anything else?
This is a very good question, and we get it all the time, and I think it's actually answered in the next slide, if I'm not mistaken. No, it's 2 slides afterwards. So bear with me. I'm just like showing here again, we go back to this to show that we kind of are measuring the same thing as you would measure with a regular single cell RNA-seq. So we have that, like the whole, we did in the same time, we did single cell RNA-seq and live-seq. So you see the live-seq samples are on my right. So we have about 40 and 60 samples with a single cell RNA-seq. And when you integrate that, you get better separation of the data points because you have more data points. really most importantly here is that you have the blue dots are the live seq sample, the red dots are regular single cell RNA-seq, so they cluster together. And yeah, I think I have to skip a couple of slides that I can answer your question first and then go back here, because otherwise you have to wait too long. Yeah, that's the one. So your question was, what is the effect of the biopsies taking on the single cells? So the experiment was set up as followed. We had some fibroblasts, we poked them, and then we waited either 1 hour or 4 hours, and that regular single cell RNA-seq on them. And when you do the UMAP projections of the transcriptomes, what you see, the 0-hour cells are basically the red dots here, and you have the green dots are the 1-hour one. And then what you see here is basically this distribution. So you don't see kind of an effect of the biopsy on the cells. And when you look at potentially differently expressed genes, that's what you get. So you have that, like there are two categories. You have gene sets that get downregulated and then come back up again after 4 hours, or you have a slight spike and then they go down. So the one that spike are stress-related genes. And interestingly enough, the one that get downregulated are basically the cell cycle genes. So what happens is, because you take 1/20th of a cell of the volume, it has to replenish it to kind of keep on growing. So you get a reset of the cell cycle and then it goes on. But as we know that these things are happening, we can kind of subtract it. Is that answering your question or not entirely?
No, because you are getting a miserable effect. If the metabolism in the cell change, it's been also argued that the changes could subvert the signal you get when you actually do it in a real experiment. Right? Because you basically telling the cell to recover. And so there's probably lots and lots of pathways that's activated because of that, and they could work synergistically with
that is possible. But still, I mean, this is still a huge advantage over just having the cell killed.
I would argue differently. You can statistically deal with the killed cells, but
no,
there's inset data.
This is sorry. I have to be really. We did a lot of calculations, and you can go to a certain amount of with statistics. You get probably the you're working with averages. Even if you do single cells, you're based on it's averaging. The effect is averaged, and you cannot link two data points directly with statistics. We can agree to disagree on that point, but unfortunately, I cannot show those data yet. But we are kind of showing exactly that.
Oh, is it okay if can you hear me?
Yes, we can hear you.
Okay, yeah, I had a follow-up question to the one you were just answering. So in the experiment you showed, is great to see that, well, after one extraction, here's the effect you see over time due to that one extraction. But then, how many extractions could you actually do to the same cell before, say, it's too much? And say, how much time would you need between those extractions?
This is a good question. So you usually wait at least 4 hours to let them recover before we take another sample. And up to 24, 36 hours is possible, and then it becomes difficult to follow the individual cells.
Okay. Great. Thank you.
No problem. You're welcome.
Actually, as the last one, what's the most number of extractions you've done to one cell?
3 up until now.
Okay.
And it's not because we kill the cell, it's rather because we don't find it anymore and the culture gets overgrown.
Okay. That's great.
So the other thing is what we do is like I mentioned that in the initial paper, they did this SmartSeq tool. So what we do is now Luthor HD. that is kind of similar, or it's really made for subcellular amounts of protein of RNA. And the idea there is that you amplify your RNA first. So RNA amplification are not cDNA making. So you add a T7 promoter to your primer, and then you can amplify the RNA linearly using the T7 RNA polymerase. And then you do the transcription into cDNA, and then you amplify it. And that makes it a lot more sensitive than the other thing. So we benchmarked the kit. So we did a dilution series with total RNA. We had 10 picograms, 1.25 picograms down to 0.15 picogram of total RNA, which is roughly, I think, about 200th of a cell. And what you can see here is you have something like 2,000 genes detected at 1 million reads. And when you go up to the 10 picograms, you're about 12,000 genes detected. So this is pretty close to a full transcriptome. And the other thing is what you can see here is that if you do technical replicates, they're really kind of reproducible. If you do the same thing with the biopsies, you see a bit more of variation. So at the 1 million read, you have between 1,000 and probably 7,000 genes detected. This comes one hand from the state the cell is in, depending on when you sample the cell. If it's bursting, you have more. If it's not bursting, you have less. But it is kind of reproducible. So I did that already. Just to give you an overview of the cell lines that we have worked with so far, like you have macrophages, fibroblasts, all kinds of cancer cells. We started working on primary or iPS-derived cell lines. And usually the cancer cells, we can find them after 24 hours, 95% of the cases, we find the cell again. The more finicky cells, it goes maybe down to 70 to 80%, but they're still there. There are some limitations, so we work mainly in 2D culture because of the geometry. And there's a limit of the cell height. If it's smaller than 1 micrometer, it becomes difficult to poke because the cell, the needle is the height of the needle. And vice versa, if it becomes too big, it also becomes difficult. So one thing that we just started now, instead of RNA, we started looking at proteins. So we have a same cell proteomics workflow now. The workflow looks the following. So we do the sampling. We dispense a bit of sample we have into a buffer with certain salts in it, certain detergents to make it solubilize the proteins. We dispense some trypsin in it, alkylation reagents. We do the digestion on the slide. Then we shoot it into a mass spec. So you probably have to take an Orbitrap Astral because that's the most sensitive instrument that is around so far. And what you get, this was the very first try. Without optimization, we detect between 150 and 200 proteins. And they were kind of also, you detected the same like proteins over again from the samples. And most interesting, when we did it kind of a bit more biological, we wanted to see if you see differences between the nuclear components and the cytoplasmic. So we sampled nucleus versus cytoplasm. And what you can see here is basically when you do clustering, the nuclear samples are here and the cytoplasmic samples are kind of away. So I was speaking mainly about the sampling right now, but there are other things what you can do with the device. So we have the injection extraction needle with the pyramidal tip and the small opening, but we have also blunted pyramidal tip for printing of, for example, proteins for doing patterns, or you take away the whole pyramid and you get a cup-shaped opening for adherent measurement in isolation. And the last one is for extracting organelles. We can also go and take out specifically mitochondria or the ER and even transfer it to other cells. So what I would like to do now is I would like briefly to look into injections. So what we can do is we can deliver either into the nucleus or the cytoplasm. We can deliver DNA like plasmids or linearized double-stranded DNA, RNAs and proteins, and also small chemical compounds. we can do this targeted isolation of cells. And we can do classical AFM measurements, so we can go and measure the adhesion force of a cell. I would like to show you just this example of the injection. So you can see again here the whole setup. So the operator is marking 3 cells now. We have set the parameters, so the force, the speed, and the pressure. You can see on the top right now the pressure and the force curve. So how we inject. So it's injecting the second cell now, and then the third. And note there are some cells that were not injected at the bottom, and I think there's some at the top afterwards. Yeah, top there. And what we did here is we delivered CRISPR-Cas RNPs that are labeled with ATA550. So now we switch now to the fluorescent viewer. And you can see this light reddish hue in the nucleus. So we delivered the Cas9 protein to the nucleus. So it's about 10,000 molecules you can see here. So I don't need to introduce you to CRISPR-Cas, but so why would you do it so complicated to do genome engineering? Because what we think is that one of the main issues that people face right now is you have to get the Cas9 protein into the nucleus of a cell. So past the cell membrane, face random transport and cytosurge degradation. You have innate immunity if you have like nucleic acids, and you have to pass the nuclear membrane. And we say we just simply deliver it where it's needed into the nucleus. And so what we can do with it is basically multiplex editing. So we can target 3 proteins or 3 genes at the same time with 3 different RNPs. And in this use case, what we did is we injected a single CHO cell and then let it expand into a colony so that we have a monoclonal cell line. And the survival rate is right after the injection is about 90%, 24 hours after the injection, and almost 50% of the injected cells form monoclonal cell lines. Editing rates of these 50% that made it into a colony, 35% have the triple knockout. And why is that? We think because we exactly deliver these 3 RNPs at the correct stoichiometric ratio, and not just like random, like you do with electroporation, that increases the efficiency. What else can you do is you can also add repair templates for example, tagging proteins with the GFP. So we did that with tubulin. So you can see here the cells have a GFP positive tubulin or nuclear proteins, and we get rates between 2.5% and 7% knock-in rates, which is quite good, particularly if you do it with hard to transfect cell lines. This is available as a service, so we don't see that someone will buy an instrument just to do genome engineering. So that's why we offer it as a service. So usually we assess the project together with the client, we discuss it, do the design, the cells are picked up at your location, shipped to us, and then we do the editing and ship the cells back. And why would you do that? So it's vector-free. They are monoclonal by design, and we know exactly how much RNP we deliver to your cell. And like that, we can also avoid off-target editing, for example. Then the last thing that I would like to show you is the mechanobiology, the isolation. So we call that the FluidFM micropipette, and we can do so-called pick and place or automated adhesion force measurements. So when you have adherent cell and you have one cell that you really want, what you do is you fill the cantilever with trypsin, go to the cell you want, you give it a trypsin shower, it will start to detach. You go down with cantilever, a bit of negative pressure, and you just lift it up, and we can place it in a new valve. So that's kind of how it looks like. And as a video, this time with adherent cells, non-adherent cells. So it picked up one cell, it moves over to the deposition site, and placed it there. This is roughly live speed. You can see the second cell and then a third next to it. And that's kind of how you could go and isolate some cells. So we can, it's not a sorter, but it's an isolator. If you have really a precious cell you really want, it's probably the best way to isolate it. For the throughput, we can do 60 cells when you have cells that are in suspension, or 20 to 30 cells if they're adherent. So next one is adhesion force measurement. That's something you can do with regular AFM as well. It's kind of complicated. You have the cantilever, you glue the cell to the cantilever, and then you press down and lift up and measure how much you have to pull that it comes off again. What we do is like that. So you see the cantilever is going to that cell. We apply a bit of negative pressure. You can see now the force we are measuring, so you start lifting up. You can see now it starts crystallizing, and now it ruptured away. And this takes a couple of minutes per cell. And with that, we can really go and not only measure like 5 cells a day, but a couple of hundred. So you can really measure them and do statistics on it. It's kind of a higher throughput. Last but not least, mitochondria isolation and transplantation. So this is also done by a collaborator. So they tested basically with what kind of opening, what do you get out of a cell? So the smallest opening, you just basically get the cytoplasm. A bit bigger, you get a bit of ER sometimes and cytoplasm. And then with these special probes here and here, you can really go and target mitochondria that are fluorescent. You can put it up. You can do analysis on the mitochondria, or you can go even and put it in another cell and see the effect of a mitochondrial transplant. And I think this becomes really important in the context of cancer biology, where you have cancer-associated fibroblasts and things like that are really dependent on the mitochondria, or how active they are, and you can go back and forth and kind of take the characteristic of one cell and transfer it to another. So last but not least, so how do people usually work with us? So what we like to do is, particularly with the academic client, they come with us with their question. We do a so-called proof of concept experiment. So we boil it down to a minimal viable experiment to provide them with data that they can apply for a grant. We offer also leasing or rent to buy, or the purchase of an Omnium. Cell line engineering we offer as a service. And I think soon we will be able to offer LifeSeq as a service too. We're building the lab right now. And then you can come and say, okay, I really want to have this drug tested on that cell line, and I want to have to connect the data points, and we will do it for you. Briefly about us. So we are about 40 employees. There are 170 publications out there. Most of them are still with the older device. There are 140 installations, and right now, I think 30 or so are the newer device, and we start really now pushing also into the US and Canada market to place an Omnium here. And with that, I would like to thank you, and I'm really happy to take more questions and kind of foster a discussion.